Welcome everyone. Today I am going to talk about chromatin organization. It's a very simple small topic. We just need to know about the nucleosome-solenoid model in this. First of all, the basics. The chromatin fibers are nothing but thin DNA molecules which are packed together. Hence, it can be also known as packaging of DNA. Many models have been proposed for explaining such phenomenon. All these models have some common few points. The first one, packaging of very thin and long DNA molecules into unit length fiber. We will know in further slides about 10 mm fiber or 30 mm fiber. Next is the production of thick fibers from the thin DNA molecules, and the last one is the beads on a string appearance of the chromatin fibers. Of all these models, the nucleosome-solenoid model of chromatin fiber is universally accepted. It was proposed by Kornberg and Thomas in 1974. According to this model, chromatin is made up of repeating units called as nucleosomes, which are 11 nanometers in diameter and 6 nanometers in height. It is composed of a nucleosome core, a linker DNA, and a molecule of H1 histone and other associated proteins, which are essential for stabilizing the entire structure. On an average, a nucleosome consists of about approximately 200 base pairs of DNA. The length of DNA associated with each nucleosome core is affected by the H1 histone and the other chromosomal proteins. That is, the 200 base pair DNA, which is the length of DNA associated with any nucleosome core, is always controlled by H1 histone, and the amount of DNA found around a nucleosome core is always controlled by the H1 histone. Nucleosome core, the next part is, it is an histone aptamer consisting of two molecules each of H2A, H2B, H3 and H4 histone proteins. They together form an aptamer. In addition to this, 146 base pair long DNA is wound around this histone aptamer, and this DNA is nuclease resistant, means it does not go nuclease activity. Next one is linker DNA. The size of the linker DNA varies between 8 base pairs to 114 base pairs depending upon the species. This linker DNA forms the string part in this model, while the nucleosome core forms the beaded part, and this linker DNA is nuclease susceptible. It can go nuclease activity, it can be degraded by nuclease enzyme. Next one is H1 histone protein. Each nucleosome in an average consists of a single H1 histone, and this histone is always found outside the nucleosome core. The degradation of H1 histone does not affect the core, and hence it can be concluded that it is present outside the nucleosome core. H1 histone is present at the entry and exit points. I repeat, H1 histone is present at the entry and exit points of the DNA and plays a role in sealing of DNA. Now other chromosomal proteins, both the linker DNA and nucleosomes are associated with other chromosomal proteins. In a chromatin, a nucleosome is about 110 angstrom in diameter. Each nucleosome stack over one another without any detectable DNA and produce a thick 100 angstrom chromatin fiber, which is also called as 10 nm fiber, that is 10 nanometer fiber. Further, this 10 nanometer fiber undergoes supercoiling to give 300 angstrom fiber, which is also called as 30 nanometer fiber. It is just a simple conversion. The other chromosomal proteins are required to stabilize this structure and to produce the solenoid organization of the nucleosome. It was pretty easy. Just revise a bit. A nucleosome-solenoid model given by Kornberg and Thomas in 1974. There are few parts in them. The first one is nucleosome core, which consists of an octamer of histone proteins, namely H2A, H2B, H3 and H4. A 146 base pair DNA is wound to it. The second one is linker DNA. The linker DNA is nothing but the attachment of nucleosome cores between the two at nucleosome cores. The attachment, it is called as linker DNA. Okay, this is from 8 base pairs to 114 base pairs according to the species. Okay. Next one is H1 histone. On an average, a single H1 histone is present around each nucleosome core. It is present outside the nucleosome core. It is present at the entry and exit point of the DNA, or it seals the DNA. And the last one are other chromosomal proteins, and in which we saw about 10 nanometer fiber and 30 nanometer fiber, and they are used to stabilize the whole structure. Okay, and this is the diagram showing histone octamers and the H1 histone present outside the octamer at the sealing and entry points, and this is the linker DNA. And this slide shows about a 13 nanometer fiber which is formed due to the coiling or supercoiling of 10 nanometer DNA. Pretty easy. These are the references. Once again, I used Genetics by B.D. Singh, Cell and Molecular Biology by P.K. Gupta, and Principles of Genetics by Snustad. Thank you.